how we're hoping to do it, why we're thinking TopMed will be a useful reference panel to use.
So Luke, 1 second. So you said slide 2. So the slide set didn't change here. The one that you're sharing.
Oh, let's see here. Let's see here. Sharing. Let me see if I can redo this here. Okay, is that?
Yeah, that looks good. Yes.
Is that now
slide 2?
Slide 2 now? Okay.
Yes, perfect. Thanks.
Okay. So the things that we'd like to do is describe our hopes here for the TopMed reference panel, why we think it's probably useful as opposed to other reference panels to do. We would also think that this might be of interest to the larger PGC community, and so maybe talk about how that might become available when we do the imputation, how we could get those imputed data sets out to other folks if they're interested. And really, I'd like to stress that for today, we have not yet started this, but it's going to take some time. A lot of data, those are huge reference panels, and we would like to get input now from all of you. And on the QC steps, best practices, how best to go about this, and how best to maybe share the imputed data after we do it. And so that's really what we want to get out of this today, is to try to get feedback at this point before we actually run it all. So that's the hope. And so at any point, feel free to jump in, ask questions, give comments, feedback. Also, my email is at the end on the last slide. So if you have thoughts, feel free to send me those via email afterwards. Okay, so what we're interested in, kind of largely in the Keller lab, are using GREML-based approaches to explore heritability. And we're interested in imputation because we'd like to see the kind of relative contribution of common and rare variants. That figure on slide number 3, that's obviously not PGC data, it's from the UK Biobank, but those bars represent different MAF and LD partitions. And just as an example of the things that we'd like to be doing with the PGC data is to partition the heritability and get a sense of the contribution of variants throughout the allele frequency spectrum, rare versus common, and et cetera. However, to do that, it really helps to have the largest and kind of most diverse imputation panel so that we can get the most information on the variants on the rarer end of the spectrum there. So that's primarily our interest right now. But I think for the kind of larger PGC community, there's potentially interest in this as well, whether it's GWAS or burden tests, that kind of a thing. I know that as far as I'm aware, the published PGC GWAS have used the 1,000 genomes phase 3 data for imputation, which is great, but it is then kind of restricted towards the more common end of the spectrum. So, possibly this using this larger, more diverse panel would get at some of these rarer variants, and that might be of interest to everyone else as well. So moving to slide 5 now. So what are the panels out there? I think probably everyone is familiar with this, but just to briefly overview it, the 1,000 genomes phase 3, it's got a great representation across worldwide populations, but it is relatively small. So we can get mostly more common variants as opposed to the very rare stuff. Currently, the kind of the big one out there is the Haplotype Reference Consortium. So this is a large consortium. It pulled lots of different studies together. It has nearly 40 million SNPs in it. It's also the default for the Michigan and Sanger imputation servers, if you're familiar with those. It's mostly European, and some of it anyway is available through EGA. And I'm not sure, maybe Stefan, you could clarify, but this may or may not be possible to implement on LISA with the Ricopili pipeline. I think that was in the works. So that's a great reference as well. And with that larger HRC data set, it results in better imputation at the rare end of the spectrum. So from the McCarthy et al. Paper, these are the numbers here. So using the 1,000 genomes reference panel at an MAF of 0.1%, the average R squared is 0.36 compared to the HRC as a reference panel, where the average R squared is 0.64. So that increase is obviously getting higher quality at the rare end of the spectrum.
Maybe I jump in directly, because there was a question to my side. So yes, indeed, this publicly available subset of EGA with these 27,000 subjects is already implemented into the pipeline on LISA and at Broad. So it's on both places you can technically use it. I use it all the time because I have the permission. I didn't get like the full answer from EGA yet. So with this question, if somebody else gets the permission to use those, can I share it then on this computer cluster with these forks? It's like similar what we do with dbGaP. So with dbGaP, we do this, if somebody shows us the permission to use the dbGaP dataset, we give them access to the specific dbGaP dataset, so they don't have to re-download this thing. And obviously, we don't want that the people download like 20 different versions of the same EGA HRC reference in there. That's obviously what we do not want to do. So I didn't share it like freely yet. So with really people that I really trust a lot, I think, which I consider basically being in the same group. I said like, okay, if you have the permission, we are in the same analysis group, and I think EGA is fine with this. I think we have to find really the solution if we can really share this like PGC wide as soon as they give us just this one email or so. It's in the works. So if somebody wants to do this, to bug EGA, I'm constantly happy to introduce it.
Yeah, but I can bring it up with you, and it seems like a very reasonable thing, and we can go to the top of the food chain and see if we can get an answer.
Yeah, perfect, wonderful. So and to be clear, so HRC actually indeed increased the quality of our imputation. So that's totally confirmed here with our PGC data.
Great. And so hopefully, maybe something like that arrangement with the permissions through dbGaP could also be implemented with TopMed if that becomes an interest for implementing on LISA or the Broad servers or that kind of thing as well. TopMed is through dbGaP. So if you go to slide 6 now, there are a lot of links on there. If people aren't familiar with TopMed, there are lots of links. I should also say that we're not actually a part of TopMed in any way. We just want to use their data. But there's lots of information in all of those links. So why go to TopMed? The reason is that it's probably the most diverse. It's at least going to be eventually the largest data set available, at least in the foreseeable future. Their goal is over 100,000 whole genome sequences really drawn from a wide range of cohorts. So that's a really nice feature. While the HRC is primarily European, this is going to have a much broader representation. And the other nice thing about it is that it's basically the same sequencing approach across everything. So they're targeting for 30x coverage, all the same methods, different sequencing centers, but they're applying these very similar QC and post-processing approaches. So it should be very high quality data. So is Santi on the call? Did he make it on today?
Yeah, I'm here. Can you hear me?
Okay. Yes. So maybe I should also just introduce Santi. So Santi is in Gonzalo Abaces's lab at Michigan, and he's worked with these data much more than me. And so I'd like just everybody to meet him, and hopefully he can fill in gaps in my knowledge as well.
Sure, no problem.
Yeah, and so one thing he just sent me this as well. So at very rare variants, so an MAF of 10 to the -4th, they are getting kind of on average R-squareds of 0.6 to 0.7. So again, getting kind of a boost in imputation quality with TopMed as opposed to others.
Can I shortly ask what imputation R-squared means here?
Santi, can you tackle that one?
So could you repeat the question once? My dog was barking, so I got a bit distracted.
Yes. The question is like, what does imputation R-squared means here? Is this comparable to the info score, or is this actual R-squared between imputed and hidden or genotypes?
This is the actual R-square. So for this, we actually like took some samples from the 1,000 genomes, and we tried to mimic like a typical GWAS study by like, masking all sites apart from those found a common chip, and then we imputed them back and just calculated the real Pearson correlation.
Okay, thanks.
And Santi, can I ask also, is that what was the size of the reference panel? Was that the entire TopMed that you have, or what?
Yeah, so the TopMed so currently the freeze that's been released, that was supposed to have around 65,000 samples. On the imputation server, I think we have around 60,000 samples, 60. Around 5,000 of them did not have permissions. And I think, and so these results are based on the panel that's going to be on the server.
So that's substantially bigger than what we'll have access to through dbGaP.
On dbGaP, it's around 50,000. Is that correct?
No, there's 50,000 total, but you only can get access to whatever Luke has in here because a lot of them are specific to specific diseases.
I see.
Which is a total waste of money, but.
So maybe moving to slide 7, there should be some information on, there we go. So slide 7 shows the numbers that we actually have in hand. So we've gotten dbGaP permission. The accession number is there. I think anybody can request this. The subset that we got approval for is the sort of general medical biomedical consent groups. There are some individuals that have specific phenotype requirements, heart, lung, blood related phenotypes because it's through the NHLBI. That's obviously doesn't apply to PGC data. But anyway, in the subset that we have access to, it's freeze 4 of TopMed. There are about 14,000 total. And in that, there are over 210 million variants, so SNPs and indels, that are called in that data set. That table at the bottom has the individual studies, and it's just in short codes. But the individual studies and the numbers, and then based on the released TopMed sample QC file, the numbers that pass all of their QC metrics, and then those that fail. The ones that pass anyway, that's something around 13, a little shy of 14,000 individuals. So I guess getting back to sample size, this is about the sample size that we have access to through dbGaP, which is obviously smaller than, I guess, the sample size that Santi was talking about with on their servers and the imputation server. But as far as I'm aware, this is about the biggest sample size we can access to sort of through these general use agreements to move on to, say, LISA for PGC imputation. So any questions so far with that? No? Okay, so moving on, slide 8. Again, sort of after just introducing that data set, what we want is for these rarer kind of frequency variants and the more diverse reference panel for these GWAS and architecture studies. And at this point, I want to emphasize too that while we're going to be doing this, we have approval for the bipolar depression and schizophrenia data sets. Maybe the imputed data could be released back to the PGC, and hopefully that could be something that would be of use to all of you as well. Or at the very least, maybe come up with sort of a defined set of procedures to apply to these kinds of data sets if folks have other other PGC data sets that they're interested in. And again, I'd like to emphasize that if you have any feedback, now would be great. We'd love to get your input at this stage. Okay, so slide 9 is just a quick steps that we'll be taking. We're going to use the phased TopMed data as the reference panel. Obviously, we need to QC the PGC data sets, phase those PGC data sets, run the imputation, and then do all the sort of post-processing, the filtering, and et cetera. So moving on to the details on slide 10. Okay, so there are a couple of things with the TopMed data set. Right now we have about 14 in hand. We have 14,000 individuals in the data set, but these are currently unphased. It's possible that in the future we'll be able to get the phased VCFs directly. We've been talking with Santi and Gonzalo about this. So dbGaP should be able to release the phased data that somewhere between, and Santi, correct me if I'm wrong here, somewhere between Michigan releasing the phased data to dbGaP and dbGaP releasing it to, I guess, everyone else. Unphased.
So I kind of forget. So what did Gonzalo say to that, like about having the, like subsetting the samples that you guys have access to directly from Michigan?
Yeah, so it seemed like he thought it might be better if we went through dbGaP on this, and so he was trying to contact dbGaP again to get them.
Oh, right, yeah, right. Okay, yeah. So I think if DB, honestly, like I don't know, I don't know how long dbGaP is going to actually take to get the phased haplotypes, because Gonzalo said that he knows that the data is with them. It's just that they don't have a download link for that. So it's like one of those things that if they wanted, they could do it really fast. But if not, yeah. So another point I wanted to make over here. So Christian Fuchsberger, who's the person behind the imputation server, they have been working on APIs. And that is specifically, as far as I understand, it's for cases like this where, for example, the PGC data which cannot be uploaded on the server. So it will be there will be APIs, so the data, the GWAS data stays on your side, and the APIs will pretty much call and somehow have the imputation done on the server. Just a small suggestion, it might be good to ask Christian once about what exactly what exactly those APIs for. But I think it was for reason something like this, where someone cannot upload the data on the server.
Okay, thanks.
So, Santi, obviously that's not done yet. And my guess is that even when it's done, it's going to be really slow compared to being able to have the data on the server. Is that right?
I don't think so, because it will be one of those things that's mainly to kind of, go around the data agreement issues, because sometimes people cannot upload the data on the server because they're not allowed to, but they still want to access a server that's going to stay behind a firewall. So this will be something in the middle where neither of you get access, you don't get access to the actual TopMed panel, and whereas on the server side doesn't get access to your data. But again, like I said, I'm not exactly sure what is the purpose of the API, but Christian might be the best person to ask about this, but just an alternative, minor suggestion.
Okay, yeah, it's something that I can follow up with at least. So thank you. Yeah, so I guess in the meantime, though, we don't have the phase data. If we can get that, hopefully that would happen soon. But in the meantime, I think we're going to keep going with doing it ourselves.
Okay, yeah, and I can help you with that in case, I can share with you the scripts that we used for the TopMed full panel. Okay,
yeah, that would be great. Yeah, and so I guess at this point on slide 11, I guess it's kind of a open question. Then what might we want to do as far as QC before we phase the TopMed panel ourselves? The release data has been filtered to some degree. I've listed the kind of some brief approaches that they've taken, and so to some degree it's been filtered, but there are also lots of other metrics that we could filter on if folks think that that would be useful, maybe Santi can provide additional kind of guidance on how they did it before. On the next slide, slide 12, I've just presented a couple of the just distributions of some of these metrics. The depth, for instance, Hardy-Weinberg p-values, quality scores, those kinds of things. At this point, I think it would probably be best just to follow probably what Michigan has done. And so with all of these possible metrics, maybe just essentially directly following them. And so if there's any other input that folks would have to say on this, I'd certainly be open to applying other filters as well.
So yeah, I might just include Hayun in future conversations about the QC part, because he was the main person who did the QC before any phasing was done. But few points I would want to mention is that the top data in general, we trust it a lot because it's 30x. That's almost gold standard sequencing. So in general, I think Gonzalez also said to kind of always go easy on the QC and keep as much information as you can, because it's trustworthy information.
Okay. Well, that makes it simpler.
Yeah, and the other thing is if the dbGaP data that's been uploaded online and that's already been filtered, then on my side I can tell that we did not do any other filtering after Hayun's filtering and before our phasing. So apart from removing the singletons, we phased the singletons, but eventually we removed them before creating the final panel.
Okay.
So, and we did not do any other QC apart from what Hayun did during his variant calling.
Okay. Well, that sounds good. I guess if there are any other thoughts from anyone else on the call, that certainly welcome to take suggestions. But that sounds good. That's easy. And then for the phasing itself, I know Santi has done this, or someone there at Michigan has done this. Yeah, and essentially, I figured we'll just take their same approach. I know that they used Eagle as opposed to Shape It. Seems like at these sample sizes and the scale of data, that's going to be the best option. We'll do that here on our servers here at Colorado. And then once we get the reference panels kind of finalized, we'll move those to Lisa, at least to do the actual imputation. The sizes at this point, I don't think will be much trouble. Currently, everything that we downloaded from dbGaP is roughly about 500 gigs. The Minimac 3 VCF format is I think more efficient, so it should be maybe a quarter of that. So at this point, I don't think size is going to become an issue. But anyway, those are just things that we're thinking about at this stage. Yeah, and then so we'll move that to Lisa, and then it's kind of on the GWAS data side, what kinds of QC to do. We've already applied. We've done some of these steps on the PGC data side, just applying the standard default Ricopili parameters for all of this. So for instance, slide 14 now, we've QC'd the depression, schizophrenia, and bipolar data sets using the standard, just the default parameters for all of these things, which I believe is what was used in the published GWAS papers. So I mean, at this point, I think that's probably a good strategy to go with. Certainly we can, these, I just copied out the table of options from the RP pipeline. We could do anything else here if there's a reason people would like to see something else done. But at this point, I guess we'll move ahead with that.
So one question here. So because you said you applied those filters, so if you, I assume you got access to the data through PGC deck, right?
PGC deck.
PGC deck is our sharing of data sets. So, or how did you get access to the raw genotypes from PGC?
So on my account in Lisa, once I got access, I got these README files that had a link to the.
Exactly. Okay, yeah. So that's what I mean. So that's the system is called PGC deck, like data access.
Sorry.
No problem. So but on these data sets, I think so far we haven't shared any pre-QC data. So I think there should not be a difference if you apply these filters.
Okay, good.
I'm not sure.
Let's hope. I don't think many sites would have been removed anyway out of this. So yeah, I figured, well, I'll just do basically exactly what the published works have done so far. So I ran through the RP pipeline with the QC and everything there.
Okay, perfect. So, but it should. I mean, let me know if you see really differences, because if we did anything additionally, so we actually. So for some data sets, we made the threshold stricter, or we did just some additional filtering, blah, 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 etc. So, but it should never be less than what you see on this list. So that's why I would be very surprised if you apply these filters that you still see anything like dropping out, any individuals or SNPs or so. But let me know if I'm wrong here. So.
Okay. Yeah, I can go back and look, but I imagine not.
Hello. Aren't you talking about applying the filters to the imputed genotypes, not the ones that you're getting from PGC?
So at this point, this would be the PGC data to then impute. Yeah. Okay, so slide 15 now. To go forward with the phasing of the PGC data itself, so the array data from PGC. I've run this through ShapeIt2 on Lisa. So I know Rick Apili now applies Eagle. It looked like it's now the default, but I had done this a little bit previously, and it was still with ShapeIt2. So 1,000 genomes as the reference panel for the haplotypes, and the default parameters for that as well. So again, sort of, I think, pretty standard for what PGC has done in the past. I don't know that anything would need to be changed, but I wanted to open up that possibility anyway to all of you. Also, it really finishes pretty quickly, and at this point, the sizes aren't really a big concern for the files. Yeah. Any comments at this stage? No? Okay.
So you imputed basically all major depression, schizophrenia, and bipolar data sets that you could get on Lisa?
So at this point, I have not done the TopMed imputation yet. This is just.
Of course, I understand. So you did 1,000 genomes phase 3 imputation.
Yeah, I have done that with 1,000 genomes.
But with all these data sets, so with like 90 different data sets?
Well,
or did you do some test rounds?
So what I've done so far is just with I guess the primarily European data sets. I don't know if there were additional ones. I think schizophrenia also had another Asian ancestry sample that I have not done yet. But yeah.
Sorry, did you use the haplotype reference panel to impute or just to phase? I mean, to me, it wouldn't make sense to impute if you're going to use a different imputation panel later. But you might use it just for phasing.
Yeah, this was just for phasing at this stage.
Right, yeah.
Okay, so this is the only phasing. Okay, I understand.
Yeah. But I guess another question though is, once we phase TopMed, would it be better to go back and rephase the PGC data using the TopMed as the phasing reference pre-imputation?
How many GWAS samples did you say that are going to be there in PGC eventually?
Depending on the data set, it's like 30 to 60, I believe, for these data sets, in that ballpark. 30 to 60,000.
And they're all no, yeah. Then they're homogeneous, like the same ancestry.
Some of them, yeah. There's at least one schizophrenia data set of, I believe, Asian ancestry, and there might be others at this point. But
yeah, but they're distributed over many different data sets. So these are a plethora of different genotyping platforms, for example. So it's definitely
And the TopMed panel that's available from the dbGaP, that also has more or less homogeneous ancestries, right? There's enough Asian and African samples.
The TopMed panel, I believe, is quite diverse. It has everything: European, African American, Hispanic, I believe East Asian samples as well.
So in general, with around 30,000 GWAS samples, I would suggest just to phase them without a reference panel, because you have enough samples that, you know, they can get enough information from each other. These things often fail, or like they don't turn out to be the most ideal way if you have mixed ancestries and they're not evenly distributed. For example, if you have only 5% of the samples which are African American and the rest are European, then these approaches are going to be kind of bad for the African American samples, because those haplotypes would get kind of biased towards the European haplotypes. But if you think that the PGC data is like, you know, like uniformly distributed, like quite uniform in terms of the populations, like all of them have more or less equal representation, then yeah, you can do them just by themselves. But if not, then then it might be better to like use a TopMed panel as a reference during the phasing.
Wait a second, but as I just stated, these are coming from very many different genotyping platforms, and I don't think we should phase.
Okay. If there are different genotyping platforms, then I think then surely
from many different. Every genotyping platform that you have seen so far is present in PGC.
I see. Yeah, then a panel is the bit. Yeah.
And some of these data sets are, so I totally agree. We have data sets, we have single data sets that you would phase and impute together that have like sample sizes of like around 10,000 or so. There we definitely do not need the reference for the phasing algorithm. But we have a lot of data sets that have like only 100 or 200 or so. That's actually
then I think the phasing, like using a panel is the ideal way to go. I agree.
Okay, so at this stage, then, kind of the approach that I've outlined here on slide 15, does it seem reasonable to move ahead then to use these phased data for the imputation itself?
I think you'd be better off phasing with your reference panel you're going to use, especially if you're looking for rare variants. That would be my feeling.
Okay.
I can't see what's to gain by phasing with one reference panel, then imputing with a different one.
Okay, yeah. I think that'll be easy to do.
And that's what you were saying you'll do, right? You're going to use Eagle or Shape It to phase with the TopMed panel, and then use the TopMed panel to impute it, right?
Yeah.
Okay.
Is that, David, Curtis, that was what you were suggesting, correct? Yeah. Okay. Okay. Great. Well, yeah, I can do that. That sounds good. Okay. So then slide 16, the actual imputation step. So we'll use Minimac, and it's nice Santi's on the call because this is he's been kind of leading this development. I have a couple of questions here. For this scale of data, I'm not sure how to best go about it. You know, imputing everyone at once, or within groups. You know, for instance, the schizophrenia data set at least does have some European compared to Asian samples, I believe. You know, the imputation itself, I know, is done on a per haplotype basis, so I don't think there's anything necessarily to be gained by running it all together. Although, you know, there's some ease of handling with lots of different files compared to, you know, a smaller number. On the other hand, the metrics like the info R-squareds, those are going to be dependent on minor allele frequencies, and so maybe how best to balance all of these questions. I'd like to ask all of you. also, the whole chromosomes, can we phase, or sorry, can we impute whole chromosomes, or should we chunk it? You know, at that point, I know that there are some higher memory nodes as well, and that we might be able to access. You know, I'm not sure if that's possible as far as just computationally, but it would certainly be easier.
So yeah, so first thing, you know, like we had the discussion last time that in general, like you said, it's good to, like, if you can impute them all together, because then you have, you know, like you work with lesser number of files. But if you have different ancestries and you believe that, you know, there's going to be quite ancestry-wise like differences, then doing them ancestry-wise is important, because then you get per ancestry R-square estimate. Now, the caveat with this is, like, you know, if there are particular ancestries that have like only say 100 samples, then your R-square estimates for those samples are just based on 100, you know, just 100 samples. So it's kind of like a balance that you have to draw, like between getting large sample sizes to calculate the R-square versus doing a stratified analysis. So I would say, like, yeah, doing them ancestry-wise is good, as long as you ensure that, you know, each of those batches have good enough strength. And regarding the chunks versus chromosomes, so in TopMed 65K, the full panel that's on the server, as well as an HRC, in general, we advise doing chunking to avoid, you know, like to avoid issues with memory. And if you use Minimac 4, Minimac 4 automatically does chunking, so you wouldn't have to do chunking. Now, that being said, your panel is going to be based out of 14,000 samples, and I think it will be fine to do chromosomes on Minimac 3 when, like, whole chromosomes, because that should, yeah, you don't even, I don't think you need like 30 to 64 GB nodes even, like each. If you run single threads on 14,000 on Minimac 3, at the most 10 gigs, I would say, yeah.
Okay, well that's helpful.
10 to 15 gigs at the most. But yeah, these are single thread jobs, and also I would also suggest using Minimac. Like, if you are going to run like multi-threaded jobs, I would suggest using Minimac 4, because the multi-threaded version is much more efficient in that one.
Okay, that's great. I wasn't actually aware that there was a 4 now, so that's good.
Yeah, we just released that like a week ago, and we have been like testing that. So yeah.
Okay, super. And maybe then a question for kind of the PGC community would be, as far as how to group these for you know, ancestry groupings. And as Stefan was talking about before, these are all from a lot of different studies, different cohorts. Do you all have suggestions on how to best go about dealing with those kind of factors? No? Just throw it all in together? All right. Okay. Well, then that's what I'll plan on doing. So slide 17 is just an example that I had done. Now this was previously something that I'd done just with 1,000 genomes as the imputation reference panel. So there are far fewer markers that were imputed here, but just to give us a sense of kind of the size of the data sets, you know, just taking chromosome 2, for instance, with, you know, over 2.5 million imputed markers, that was 54 hours, but it completed just fine as a whole, throwing all the samples in at once. The sizes, I think, is something that will become a problem. So I had previously done this, and because I was running out of room on my home directory on Lisa, I deleted the VCF files because those were huge. So I don't know the actual sizes of those, but for the filtered, and you can see the sort of the filters that I applied with minor low frequency and info R-squareds. You know, the filtered data for all 22 autosomes was 144 gigs. And that's in the bipolar data set with about almost 11 million markers and 56,000 individuals. I can only imagine that using TopMed with many, many more markers imputed, it's going to be much, much larger. So that's maybe a challenge. I'm not sure how, yeah.
Sorry to interrupt. So is there any reason why you were keeping them as Plink binary files instead of just?
Oh, it was just easy for the approaches that I was using. GCTA uses Plink binary.
Dosage files, I mean, so the imputed VCF files, like, you know, VCF zip files are going to be much, much smaller than a Plink binary file. And yeah, I do not have an estimate, but yeah, like, you know, for here, over here, just a single chromosome. Okay, so you are saying all 22 chromosomes take up 144 gigs, and that's around 10 million markers. With TopMed, if you're using the 14,000 panel, it will be higher, but it might be around to like 50 million, just a rough estimate. Do you remember how many markers are there on the TopMed 14,000? I think your mic is muted.
Sorry. About 210 million markers.
That's including singletons, I assume?
It's whatever was in those files. So yeah, I guess. And indels and everything else. So
210 million markers from 14,000?
Yeah.
That doesn't seem right to me.
It's the number of lines across all the 22 VCF files.
I'll just check with Gonzalo. I don't think they would do something like this, but it seems like they put in all the variants across the 60,000 samples. And I don't know, because I know for a fact that like our entire panel, like with singletons, is around 450 million variants. And after removing singletons, that's on the server, that's going to be around 200 million variants. So I think it might be that they put in all variants, and so majority of the variants in that data set would be monomorphs. My guess would be, around with 14,000 samples, my guess would be around, you might have around 50 to 60 million variants, you know.
Well, that would help with the sizes of the.
But that's, I mean, just like, you know, just do it on chromosome 20 to get an idea of how many non-doubletons and above are there.
Mm-hmm.
Okay. So Luke, we plan to keep the non-filtered files too, right? And is that right?
Yeah, I mean, I would like to. Again, at a certain point, it's going to come into storage issues.
Okay, and those will have as many markers as there exist in the reference panel, right? So 60 million, so about 6 times bigger than what you are showing there. So it's not ridiculously huge, right? Less than a terabyte.
Yeah, I guess. Depending on how many markers would be removed as singletons from the reference.
Yeah, and even in a VCF format, it should be like even less. So like, I mean, I understand you need the binary files, but you know, like it can be like the VCF files are the main storage, and whenever you need the Plink binary files, you just temporarily convert them. The other point is like also Plink binary files, I'm not sure, they can't convert dosage values as a continuous value, can they? I think they just look at the hard call best guess genotype. So you'd be losing some information. So depending on which association tool you are using downstream, as far as I know, most of them allow VCFs nowadays. So, and they can take in the dosage values. So unless it's like different questions that you are answering, but for usual typical association tests, most of them will work with the VCF files that come out of Minimax. Yeah,
certainly. We're kind of interested in these heritability methods.
I see. Yeah, then
takes in the binary files and I believe not VCF at this point. But certainly that's another question, is kind of what format might be useful for folks. Certainly VCF, maybe Plink binary, maybe BGen or some other format.
So BGen, I was going to suggest BGen, like if it's an association test that you are going to run again and again and you have lots of phenotypes, In that case, BGen, I would suggest BGen. If it's something that's going to be, just once or twice, then just stay with VCF. The problem is like currently in Minimac 3 or 4, we do not have the BGN support. We have been talking with the people from Oxford to like have like that support. So if you use BGen, you have to use QC tools to convert it and then. So
yeah.
So yeah, if you don't need to run multiple association tests, then just stay with VCF files.
Okay. Well, it sounds good. It maybe, and especially with fewer markers after removing singletons, maybe space won't be such an issue, which would be nice.
Yeah. I'm sure 210 million variants that's not possible on possible on 14,000 samples.
Okay, sounds good then. Okay, so on slide 18, then sort of just some questions maybe for everyone on the call. We have access to, and we're going to be imputing depression, bipolar, schizophrenia data sets. If there are other target data sets that people are interested in, or at least making scripts and those kinds of things available, or the imputed data themselves. If there is interest, if people would like to see this, perhaps filtered or not filtered, those kinds of questions. Certainly open to any input, any suggestions from the larger community here. And so slide 18 just kind of lists some of those questions that we'd like to get feedback from all of you on.
So one minor point, like I know we will talk about this in the future again when you actually run the imputations, but in Minimac 4, we have a new format called HDS. That's, it's good to keep that format turned on because that is something you can convert to anything else you want in the future. For example, things like genotype probabilities, they lose phase information. Then the dosage loses the haplotype information and things like that. But HDS is just like, haplotype based dosage. So you will get two values separated by a comma. And just a minor point to keep that turned on. That way, in future, any other format that you need to do, that's necessary and sufficient. You can get anything from that.
Okay, great. Okay, and then just slide 19 is the last slide. Thank you for your feedback, and I really would appreciate anybody's comments or thoughts on this. My email address is listed there. If you think of something, tomorrow or next week, please send it my way. If there are any other thoughts or comments.
So David, did you want to bring up the point you brought up in the chat?
Well, I think I said what I wanted to say. I think there may be some advantages in having a whole genome sequence, maybe in terms of quality and obviously in coverage. But I think also, I guess it's not going to be possible to bring in the exome sequence data that we already have alongside the TopMed stuff that you have. So I guess I see them as kind of slightly sort of complementary. I don't think this is going to pick up the sort of very rare variants with big effects, but it may pick up more of the sort of rare, quite rare variation.
I don't know. Is there a way to combine those reference panels together, or would it just be kind of two separate imputations? Or
so this is, I guess, where I can chime in again. So like we are currently working on something called like meta minimac, which would be like the approach like that we are calling that meta imputation. And the idea is like pretty much what you were saying. So if you have a single GWAS and two reference panels, ideally you would want to merge the reference panels, or like if you had access to the BAM files, you would want to jointly call and create a merged big reference panel like we did in HRC. For example, we had 20 studies and we merged them, but that's not possible always. So this is kind of a roundabout approach where you impute against each of the panels and then you merge them by a weighted combination, and those weights are chosen in a way that it gives you as much accuracy, almost as much accuracy as the merged panel. And in some cases, especially in the rarer spectrum, it gives slightly better accuracy. And so we are planning on releasing that like within the next week. It's almost done. That is something you can give a shot in the future. like if you have like a separate reference panel, and you want to combine them with TopMed?
Yeah, I think David's point is that, if we have some cases, they're likely to be enriched with highly penetrant, very rare variants, which are less likely to exist in a control sample. But the problem, as I see it, is just pragmatic, David. It's just that I think it's gonna be a real mess to try to merge, exomic sequencing with the whole genome TopMed sequencing.
I mean, I'm doing that anyway with the exomes at the moment. I mean, I'm struggling with the resources, but so I mean, they can sort of carry on in parallel, and then maybe we can, see, compare what kind of results we get from the two complementary approaches.
Yeah.
And if you want to swap notes about these kind of things, about bring things down into chunks, using the archives, scripts, and so on. I've been struggling with this myself, so I'm perfectly happy. I think maybe offline to take that forward.
That'd be wonderful. Yeah, we'll take all the help we can get.
And Santi, can I also ask just briefly, so you guys are able to access the entire TopMed as a reference panel, and that's just because you're part of the TopMed, or is there some way that we could also do that?
Yeah, I have access because I did the phasing and I created the reference panel. Yeah, but yeah, apart from that, I don't know. Like Gonzalo would be the best person in terms of.
Okay.
So just one question: exome sequencing, is that something that is already done and available, or is that something that you guys are deciding whether to do based on funding?
Yeah, I mean, sort of in the process of doing it. I did it on some, and then I've kind of enlarged the number of exome sequences available a bit, and then I'm kind of redoing it, producing the phase reference panel and then imputing. So that's kind of going ahead. But that's only in the schizophrenia sample. So that's an approved sub-analysis of the PGC schizophrenia 2 sample. And
sorry, yeah, and how many samples would this be in the exome sequence?
Right, so the exome, it's about 11,000 subjects, about 5,000 with schizophrenia. And yeah.
And the rest are just samples?
The rest are controls, Swedish controls.
I would still like this. I mean, I would still like, I would still use this somehow. Like 5,000 samples, exome sequencing is still some information. Yeah, that's still a good amount of samples that, that shouldn't be.
But the other thing that's happening is there's more exomes coming online all the time. I mean, so there's a schema group, and they're going to have several thousands of schizophrenia and I think also bipolar exomes, and they're sort of overlapping but separate from the PGC. And so, maybe we should bear in mind whether there's some way we can bring all of that together.
I see. Yeah. But yeah, I'm saying like, even 5,000 schizophrenia samples, just exome sequencing is still like, it's some good information.
Well,
yeah, well, I'm happy to, to share where I've got so far. Have you guys got a sort of dedicated resource for this? Because one of the things that's been holding me back is I haven't got any additional enhanced access to LISA resources, and so I'm finding things go kind of quite slowly. So if you were able to purchase a larger resource, then things would get done much more quickly, I think.
Yeah, we don't have any special allocation or anything like that. Matt Keller had bought one of the high memory nodes on LISA, and I don't know if we get priority now to that.
That resource is available to the entire PGC. I think, we have priority access, but we don't use it all that much. So David, I think all you need to do is to write Danielle. I mean, it's a slow node, CPU wise. So if it's CPU heavy, you're probably better off just going through the queue. But if it's a high memory thing that you need, then that might be a good way to go.
Yeah, no, I wasn't thinking of that particular node, but just in general getting access to more processing power. But yeah, we can maybe discuss that. And,
yeah.
I mean, maybe I'm doing things all wrong, but I'm finding it's taking quite a time.
So sorry to interrupt now, but we have only 1 minute left, and as I said before, I have to stop in exactly 1 minute. So first of all, I want to thank Luke and Matt here to present this absolutely thrilling stuff, and it's no wonder that there's a lot of discussion here already. Of course, it's great to have Santi also in here, so it might be probably a good idea to actually share Santi's email, or maybe Luke, you want to prepare like a working group with David and Santi and a couple of others, so that we bring together the. And I'm totally interested in, because this is like also PGC core work, if this really works out. So let's try to get in touch, or like to keep in touch, and to have like a core unit who is discussing this. And again, also everybody here on the call, we had like many participants is again invited to join us here and to actually contact Luke with ideas, as he said here before. Sorry to not give everybody the possibility to ask questions here, but I think we had already a very vivid discussion. And again, thanks to you guys, and thanks to everybody else. And if you have more questions, just email us.
Okay.
Right. So I have to stop.
Thank you.
Bye bye.
Okay. Bye bye.
Cheers. Bye.